Microtubule is one of the second most important cytoskeleton structure after the actin. So we'll discuss in this lecture about the microtubules and the various motor protein which are usually involved in the movement across the microtubule. So microtubule basically it is a rigid hollow cylindrical and it is diameter ranges of the 25 nanometer, and it is dynamic structure just like that of the actin, which means the assembly and the disassembly of the microtubule assembly and the disassembly of the microtubules take place again and again just like the actin molecule, and basically its role or its function is providing in the shell shape and also in the movement of the cell, or it could be a intercellular movement of the transport of the organelle from the one place to the another place, or from the transport of the vesicle from the one place to the another, and also the separation of the chromosome during the mitosis is also the one of the important functions of these microtubules. And basically they are made up of single type of the protein which is known as the tubulin protein, but instead of the single monomeric, there are the heterodimeric molecules are present. It means the two types of the tubulin, that is the alpha and the beta tubulin, they form together or they polymerize together to form the dimer, and this dimer then polymerizes to form the microtubule. Okay, while in the actin, what was the case? The actin is just made up of single monomer, which was the G actin. Okay, and several MAPs. MAPs means microtubule associated proteins are basically involved, which are mainly responsible for the regulation of the synthesis and the disassembly of the microtubule. Now we'll discuss about the structure and the organization. The structure, as we have discussed, that it is made up of the globular protein, which is known as the tubulin, and basically it is of the 55 kilodalton polypeptide, and they are of the two type, the alpha and the beta tubulin. The alpha tubulin basically it is coded by the alpha tubulin protein coded by the variety of the gene, and the six genes are responsible for the coding of the alpha tubulin, while the seven genes are responsible for the coding of the beta tubulin. Now, what happened? These alpha beta tubulin they arrange like this. Okay, this is the beta and this is the alpha. So it formed the dimer. Now, after the formation of the dimer, it formed a protofilament, that is it composed of the linear chain of these dimer, that is the beta alpha beta alpha beta alpha. In this way the structure, the linear arrangement of these dimer are formed, which is known as the protofilament. Okay, usually it consists of the 13 protofilament, and all these protofilament then wrap around themselves. It is present in the form of the sheet, and when like in the sheet, okay, in this manner. And afterward, what happened? It form a wrap around themselves around a hollow core and forming the structure like this. Okay, so this is the basically the microtubule structure. It's look like this consist of the protofilament consisting of the 13 protofilament units. Okay, and besides the alpha and the beta tubulin, the gamma tubulin are also present, and they are not responsible for the formation of the microtubule. Instead, they initiate the microtubule assembly, and they are basically present in the centrosome region and are responsible for the initiation of the microtubule, and they have the certain pericentriole, or we can say they are present in the pericentriolar region which are present around the centriole. So Due to this arrangement of this protofilament, that is, the dimer are arranging in the linear manner in the form of the protofilament, and which afterward the polymerization of the protofilament form the microtubule. And due to this arrangement, they have a distinct edge like that of the actin molecule. They also have the minus N as well as the plus N. Now, due to this polarity, what happened? This polarity is basically responsible, or it helps in the direction of the various motor protein along the microtubule. So they are the dynamic structure, and they are regulated by the um, reorganization and the organization, the disorganization against and again. That is why they are dynamic structure. They are not rigid or the static structure. Now, what happened for the polymerization? Some form of the ATP or the GTP molecules are required. So in this case, the alpha and the beta tubulin, they are bind to the GTP molecule, that is the guanosine triophosphate. Okay, guanosine triphosphate. While in the case of the ATP, what happened? In the actin, the ATP was bind to the G actin monomer. So in this case, the alpha and the beta tubulin, the both are bind with the GTP, while the beta tubulin, which is bind to the GTP, they are undergoing the hydrolysis process very soon as the polymerization is formed. So what happened? They are hydrolyzed and forming the GDP, and the beta tubulin, which is attached to the GDP, they form the weak bond between the dimer. Now, what happened? The alpha and the beta dimer, which is attached to the GDP, they form the weak bond of association to each other, and that is why they dissociate from the filament, and they are dissociating from the microtubules, that is from the minus end of the microtubule. That is why the minus end of the microtubule, it needs to be the protected, and that is why they are anchored in the centrosome region, or we can say they are anchored in the microtubule and organizing center, which is also known as a centrosome. So what happened? The negative part of the microtubule, it is embedded in the centrosome region, and this empty C, or the we can say the centrosome, is consists of the gamma tubulin, which are responsible for the synthesis of microtubule. Now there are the variety of the microtubules are present in the different pattern. Either it could be present in the single microtubule. Single microtubules may consist of the 13 protofilament unit. Okay, like this. Okay, this is a 13 protofilament, suppose, and it is a singlet microtubule. Now, doublet microtubule means two rings of the microtubules are fused together. The first one may contain the complete protofilament, it could be a 13 protofilament, and the second ring may consist of the 10 or the 11 protofilament, and these two are the fused together. So, this is the double microtubule representation. Now, in the triplet microtubule, what happened? The three microtubule rings are fused together, and it and again the case may be the one of the microtubule of the triplet cases contain the full 13 filament, it could 10 or 11, like this way. So in this way the different microtubule patterns are present. So the structure, we will the structure of the microtubule will be clear from this beautiful diagram, as we have discussed that it is made up of the alpha and the beta tubulin, or we can say the backbone of the microtubule is the dimer. So the dimer of the beta and the alpha, in this manner. Okay, so they are forming a sheet around a hollow cone, or we can say at a hollow core. In this way, it form a sheet. The alpha tubulin is there and the beta tubulin, and the diameter of this ranging from the 25 nanometer. And in the 3D dimensional structure, we can see that the beta tubulin is both the tubulin are attached with the GTP. Okay, GTP is there, but beta tubulin, which is attached to the GTP, they are prone to the hydrolysis as soon as it is attached to the microtubule. So that is why it is attached to the GTP molecule. Now, GTP play a very important role in the microtubule polymerization, and the addition of the microtubule dimers are basically from the positive end, and the dissociation is mainly from the negative end. Now, what happens as soon as the dimer, which is attached to the GTP molecules, they are added and undergo the polymerization process. Now, after the polymerization, what happens? The GTP molecule which is attached to the beta tubulin, they undergo the hydrolysis. And after the hydrolysis, what happened? The GDP molecule is formed. That is from the GTP, the GDP molecule is formed. And when the GDP molecule is formed and they are attached to the beta tubulin, they form the weak association with the alpha tubulin, and therefore this weak association of them help in the or it promote the depolymerization. That is why the dimers of the tubulin, they are dissociated from the negative end of the microtubule filament. So what happened? These minus end of the microtubule, they must be protected, and therefore they are anchored in the centrosome region. Okay, centrosome region to prevent the rapid dissociation of the tubulin molecule. Now, the dynamic instability of the microtubule. Dynamic instability, it is basically a transition phase, or we can say switching between the phase of the slow growing and the rapid shortening of the microtubule. And the slow growing is basically called as the rescue, while the rapid shortening or the shrinkage is known as a catastrophe. Okay, now what happened and what lead to the dynamic instability? As soon as the GTP bind tubulin molecules or dimer, they are added to the positive end of the microtubule. Now what happened? It provide us some gap, or some cap is provided by the GTP. Now, what happens if the cap is provided by the GTP? So, as long as that cap is present, they are not undergo the hydrolysis. But if the hydrolysis is going to happen, the GTP is converted again to the GTP, and the shrinkage of the microtubule, that is the removal of the dimer molecule from the microtubules take place. So in this diagram we can say in the growth they are very slow, additions of the microtubule dimer, or we can say microtubule filament, because one by one they are added to the microtubules, the dimers. So the dimers are adding, and again the hydrolysis in this way. They are again hydrolysis of the GTP molecule. So what happened? They are protected by some GTP cap, as the head is protected by the GTP cap, as we can see. So what happened? As soon as this GTP is undergo the hydrolysis, this cap is removed. This cap is removed, and if this cap is removed, the dissociation of the GDP molecules along with the tetra molecule take place, and this results in the shortening of the microtubule, or this is known as the shrinkage. And the hydrolysis basically of the GDP take place at the plus end, because the negative end are usually anchored in the centrosome only. So, to protect this or to regulate this dynamic instability, the different microtubules associated protein are usually attached to the microtubule, so that to regulate both the polymerization and the depolymerization uh, in a very regulated manner. So what happened basically, the three classes of the protein are attached to the microtubule. In the first class of the protein is the polymerase. So the polymerase, when added to the microtubule, what happened? The um, additions of the dimer molecules attached with the GTP, they increases to the tenfold. As we can see earlier, there were only the one molecule at a time are added inside into the microtubule, or they are undergoing the polymerization. Now, after the addition of the polymerase, what happened? The tenfold of the dimers are added to the microtubule to increase the or to for the rapid growth of the microtubule. So, as soon as the polymerase are enzyme, the additions of the dimer are increased to the tenfold. So here the polymerase are attached to the positive end, and the addition increases, or the growth of the microtubule is increases to the many-fold times. Now the second class of the protein is the depolymerase. Now what happened if the depolymerase attached to the microtubule, the cap of this GTP is removed, so the depolymerization of this cap take place, and therefore as soon as this GTP cap is removed or the depolymerized, they are undergoing the shrinkage stage. Shrinkage stage means it it is dissociating, the dimers are dissociating from the positive end of the microtubule, and therefore responsible for the shortening of the microtubule, or we can say the catastrophic effect. Okay. And the third class of the microtubule associated protein is the clasp protein. Clasp protein basically, when added to the microtubule, it helps in the reduction of the catastrophic stage, and it promote the rescue. Rescue means again it help in the slow growing of the microtubule filament. So the clasp, when attached to the, it is protected, it protects the microtubule filament in a manner that it help in the preventing the shrinkage stage and the catastrophic stage, and help in promoting the rescue stage. And therefore, after the addition of the clasp, the growth is resumed, and the additions of the dimers with the GTP molecule, they are attached to the positive end of the microtubule. So the MAPs, or we can say the microtubule associated protein, the opposing action of these MAPs are help in regulating or the transition between the shrinkage as well as in the growth of the microtubule filament. Now we'll discuss about the intracellular organizations of the microtubule, or we can say the assembly of the microtubule. So microtubules basically, we can say they are extend from the centrosome. So this is the centrosome region, the green region in this manner. The centrosome region and microtubule are extended from the centrosome. This centrosome region is also known as a MTOC, that is microtubule organizing center. And it is the only site which is responsible for the initiation of the microtubule, because it contains the gamma tubulin. So microtubule basically it arises from the MTOS or the centrosome region, and they arises or extend outward in such a manner, we can say the positive side are present at the periphery of the cell. So the positive end present toward the periphery of the cell, while the negative end, this one is the negative end, and it is embedded inside into the centrosome. Okay, so they are arranged in this manner, and that is why that is why the minus end or the minus end is embedded or anchored in the centrosome. That is, the dissociation of the dimer molecules are protected in this manner from the minus end, while the dissociation of the dimer molecules from the positive end, they are established by the MAPs, which we have already discussed. So this is how the microtubules are organized inside into the cell. Okay, so this is basically a stage of the interphase. Interphase means no particular growth or the division taking place, and cell is duplicating its item inside the cell. Now, what happened as soon as the cell undergo the mitotic division? What happened? The two sets of the centrosome is now placed because it duplicates its material. The cell is duplicating its material in the interphase state. Now as soon as it enters into the mitotic state, the two different centrosome, or we can say that two different MTOC centers are present, and from them the microtubules are extended to the outward. Okay, in this way they are helpful in the arranging of the, or we can say the assembly of the microtubule is possible. And in the centrosome only there is some pericentral area which consists of the gamma tubulin, and which help in the uh, initiation of the microtubule. Basically, what happened in the mitosis, the microtubule, it extends from the daughter centrosome. Here it is a daughter centrosome, and it is forming the daughter centrosome, it forming the mitotic spindle, and these in spindle are necessary to separate the chromosome to the daughter cell. So this is the role basically provided by the mitotic spindle. Okay, daughter cell, one cell, daughter cell. And the microtubule basically it is present in almost all the animal cell, and while in the case of the fungi, the spindle pole bodies are present which function like that of the microtubule, or it is analogous to the microtubule only. While the plant do not have the microtubule in it, instead they have some array underlying inside into the plasma membrane which help in the synthesis or the division of the plant cell wall. Okay, so this is how the assembly of the microtubule take place, and beside their assembly in the microtubule, they are responsible for the various providing the cell shape, the cell movement, the intercellular transport of the vesicle. Now, dynein and the kinesin, there are the various motor protein are present which moves along the microtubule. Now, as we know, the microtubule basically are responsible for the various function either to the cell movement